No, I think we sort of appreciate like that you do some of the introductions, because you sometimes you say funny things that not like we do not know. Right? Well, like telling people, oh, like he used to break this, he used to break that, and
yeah, so many bad lab benches, messy.
yeah, exactly.
But we generally wait till 5 after to begin. And there's Art Palmer. How are you, Art Palmer?
I'm fine. We got our center grant submitted, so I can talk to you about it whenever you want.
Congratulations.
Yes. Oh. And Kendra, I have about 6 meetings going on at the same time, so I may be jumping in and out of your seminar. I apologize.
You're going to miss some really great stuff, but that's okay. I'll catch you up.
I hope some NYSBC data.
At the end, a little bit. We're still working on the analysis. Some of it's a little messy.
My day was complicated to begin with, but then I had a crisis this morning about an international bank transfer that had to get sorted out.
That sounds fun.
Yeah, it wasn't, actually. They don't take it so well when you try to do an international bank transfer in an account with not enough money.
Oops.
Is it in Bitcoins?
Oh, it's in real dollars. It's part of my work as executor for Barbara Lowe.
Hmm.
I didn't realize the lawyer was actually going to cash the check for his duties at the same time I was making the bank transfer.
Hmm. That's a problem. Yeah. Hmm. Yeah.
But we all got a 2-week reprieve on S10s. They were supposed to be due today. But at the end of the last week, the NIH postponed the deadline to the 15th.
Yeah, that's what I heard. Yeah. Hmm. Yeah, we're trying to get our 34 gigahertz, our Q band upgrade settle. It's still not settled. So it's the only place we can do pulse DNP right at the moment. It's Q-band.
Mhm.
Yeah. Because Sudheer has not finished his amplifier.
You can blame me, Bob.
Well, it's working. We just have to get it back up and running and get the get Michael's very nice driver system down there.
Yeah, we'll get that there soon.
But if you
Or else
didn't have them wasting time giving seminars, you could be getting it fixed.
All right. Okay. So Kong, you want to get started? It's 5 after now.
Or yeah, sure. Okay, this is what else should I say? But just give me 1 minute. Let me see. Let me copy and paste this. Yeah, I mean, good morning, good afternoon, good evening, everyone, like wherever you are. I mean, welcome to our ZOOMinar series again. As usual, I would do the usual thing that I've been doing over the past 1 year. That is like, I mean, if you have not like, if this is your first time here, feel free to subscribe to our mailing list so that you get informed about like the future seminars. And also, like if you want to look at our past recordings, they are all of them are uploaded onto our website. And of course, if you want to ask questions in the end, just use the Q&A function that you can see at the bottom. I put all the information into the chat, so you can feel free to just copy and paste and subscribe to it if you want, the mailing list. So today we have two very good speakers. I mean, the first one is Sudheer Kumar. So like, I mean, like, of course, like for us who have been in Bob's group, we all know like Sudheer very well, because like he's usually the go-to person if any of our Gyrotron breaks, and then like if we have any like microwave issues and with our probes, we usually go to him because he's, you know, he knows everything about it. And personally, it was a pleasure working with him. In fact, I was working with him on the 140 the Gyrotron. That was the first Gyrotron ever built. It was very ancient and also a lot of issues, and Sudheer gave me a great help. So it's a great pleasure listening to him today to tell us more about what he has been developing for the gyro amplifier and also the light-driven semiconductor switch, laser-driven semiconductor switch. So I will just tell you a little bit about his portfolio. So he did his he got his master in science in physics back in IIT Delhi, and after that, then he joined the group of John Philip for PhD in Swiss Plasma Center in EPFL. I mean, that's the center where you have where they are. If I'm not mistaken, like that's the group that they try to build a lot of microwave devices for plasma fusion applications. And then after that, yeah, he came to MIT and joined the group of Richard Temkin. And then after working as a postdoc for a few years, then he's now a research staff scientist at PFSC. And then of course, he maintains a very close collaboration with the Griffin Group. And very recently, he just got awarded an R1 grant. And then of course, Bob is trying to convince him to spend all the money on the gyro amplifier. But we will see how, maybe he will tell us how he's going to spend the money for the next 5 years. Welcome, Sudheer.
Thank you, Kong. Thank you for this introduction. Okay, let me start sharing my slides first. All right, can you see the slides?
Yes, it's good.
Okay, yeah. So I will not talk about how I'm going to spend the money, but rather I'll talk about our recent progress, what we have made in our group in developing microwave and THz sources and techniques for DNP-NMR and also the pulse DNP-NMR. So as Kong also mentioned, the research goal in our group is to demonstrate advanced instrumentation, microwave instrumentation for DNP-NMR. primarily developing high power and high frequency microwave sources, which are called gyrotrons and gyro amplifiers, and also pulsed microwave DNP techniques for pulsed DNP-NMR. So the outline of my talk, I'll briefly talk about the gyrotrons, the brief overview of the gyrotrons for DNP-NMR. Then I'll mention about a recently developed 527 gigahertz gyrotron at MIT for the 800 megahertz NMR spectrometer. And then I'll talk about the short pulse generation from the long pulse gyrotrons for pulse DNP, and then I'll talk about the phase stability in gyro amplifiers for pulse DNP NMR. So to give you a state of the art of the DNP gyrotrons, as you know, gyrotrons are the only microwave sources which can fulfill the power requirements for DNP-NMR in the frequency range, which is about 200 megahertz to recently developed 1.2 gigahertz NMR systems. And the matching frequency for gyrotrons in this NMR frequency range is about 100 to 800 gigahertz electron cyclotron frequency, the microwave frequency. And over the years, there have been a lot of gyrotrons developed for DNP-NMR. And at MIT, we have developed, as a lot of you know, our own set of gyrotrons. And you can see one picture on the top left here, our 250 gigahertz gyrotron. It was developed about 15 years ago and still running nicely for doing pulse the CW DNP-NMR at 380 megahertz. On the bottom left here, you see our 460 gigahertz gyrotron, which was also developed a long time ago for the 700 megahertz NMR system, and it's been in use since then. On the middle top here, you see a picture of the early version of the Bruker CPI gyrotron that was, I think, one of the first one developed at 263.5 gigahertz gyrotron. This is a fundamental harmonic operation for this gyrotron. And there are other groups which also developed their own gyrotrons, like the Fukui University gyrotron, which was developed at 460 gigahertz and now installed at Osaka University for 700 megahertz NMR. And on the right, you have a picture of the Bruker CPI 527 gyrotrons. And you know, Bruker is the main developer for these systems. And they have developed up to 593 gigahertz for the gyrotrons, and they're probably working for gyrotrons at the recently developed 1.2 gigahertz NMR system. To give you a brief overview how a gyrotron works, as you know, a gyrotron is a vacuum electron-based device, meaning that the microwaves are generated by a resonant interaction of an electron beam with the field in an open resonator cavity, which is somewhere here. So there are 3 or 4 main components to the gyrotrons. So you need an electron gun to generate that electron beam, which is operating at about 15 to 20 kV and about 200 milliamps beam current. It's about 4 kilowatt on an average beam power. So this electron gun generates an annular electron beam that is compressed through this main magnetic field and passes through a resonator cavity section here, which is an open resonator cavity. And the wave particle interaction happens in the resonator cavity, where the electron beam energy is transferred to the transverse electric fields in the resonator cavity. And those fields are generated as microwaves, and that field is converted from a TE, transverse electric mode, into a Gaussian-like beam using a set of mirrors and a launcher, which we call, in general, a mode converter, and through the window here. And you see the Gaussian-like beam generated from one of our 460 gigahertz gyrotron. It's a nice looking Gaussian beam. So the frequency goes as it's an electron cyclotron frequency. So it goes as EB over M, which is about 28 gigahertz per Tesla. So if you are operating in the fundamental mode, you can go up to like 10 or 12 Tesla with the state-of-the-art gyrotron magnets, and you can go up to 300 gigahertz if you are operating in the fundamental mode. If you operate in the second electron cyclotron harmonic, you can go up to 600 or 700 gigahertz. And this is one of the advantages operating in the higher modes. You don't have to spend a lot of money on your magnet. So you can have a standard 10, 12 Tesla magnet, and you can go up to 700 gigahertz with the second harmonic operation. And as you know, at MIT, we have been developing our own set of gyrotrons, and we recently completed our 527 gigahertz gyrotron, which operates in the second electron cyclotron harmonic. And you can see the CAD drawing here for the gyrotron. The green part is the superconducting magnet, and the gyrotron tube is in the magnet right here. And as mentioned earlier, the electron gun, which is at this end of the gyrotron, and the resonator cavity is in the center here. So the electron beam that is generated from the gun passes through this long section called beam tunnel and goes through the cavity and goes gets absorbed on the collector here, and microwaves are generated and converted to a Gaussian-like beam in the mode converter right here in this section here, and coupled through this internal waveguide to the gyrotron and comes out of the window here. So the gyrotron typical operating parameters, the voltage and current are 17 kV, 15 to 17 kV, and about 150 milliamps here. And our operating mode is a higher order transverse electric mode, TE11.2. And the Q number is what we call the axial mode number, which you can access to get the frequency tuning in your gyrotron. And the output mode at the window is directly an HE11 mode, because this gyrotron has a nice feature that it has this internal waveguide assembly that directly couples from the mirror setup to the HE11 mode in a corrugated waveguide. And one nice feature with this gyrotron we aimed was to have a frequency tuning of about 1 gigahertz. So that was the design goal. And the advantage of having a frequency tuning in your gyrotron is that if you have a nice tuning, you can sweep across the line, DNP line width, and then you can see the DNP line shape there. Also, if your NMR magnet does not have a sweep coil, you can adjust the frequency of the gyrotron to match the field on the NMR magnet. So the experiments were completed, and what I'm showing here is what we call the voltage tuning and the magnetic field tuning. So you measure the power and frequency as a function of voltage, where you fix the magnetic field to some number. And operating at about 110 milliamps of beam current, we got a maximum power of about 9.3 watts here at the frequency of 527.22 gigahertz. The frequency came really close to the 527 design number. And with the voltage tuning, we got about 350 megahertz of frequency tuning. And on the right here, the magnetic tuning, you fix your voltage to some number, 16.7 kV, and then you vary your magnetic field and measure your power and frequency. And we got about a similar performance of 9 watts of output power and 527.2 gigahertz. And the overall frequency tuning, when you combine the voltage and magnetic field tuning, is about 400 megahertz with an average power of over 4 watts over the entire frequency tuning range. So we were pretty happy with the results, and we completed the gyrotron and installed with the 800 megahertz NMR spectrometer. And we expect to increase the frequency tuning and power in future, where with the long-term operation, we can have an improved pressure in the gyrotron, and we can increase the beam current to get more power and frequency tuning. And these results were published in early 2020. So the gyrotron was installed with the 800 megahertz NMR spectrometer. Here you can see in the picture. The right side shows the gyrotron here, the green magnet. The tube is in the vertical configuration here. So we designed a corrugated waveguide transmission line that runs from the gyrotron to the NMR probe in the magnet here. And the waveguides that are designed are, it's a overmoded corrugated waveguide that is about 16 millimeter internal diameter, which runs about 4.5 meters from this section to here, including 2 miter bends. And there are tapers that goes from the gyrotron internal waveguide that is 12 to 16 here. And then the 16 goes to 8 millimeter, about 8 millimeter, which is the diameter of the waveguide in the probe. And we measured the transmission efficiency, and we found a very nice transmission efficiency of about 80%, over 80%, and the very minimum loss of about 20%. So it's an excellent transmission efficiency for the system, and the system is more or less in operation right now. And you see in the picture also Kong, Eddie, Edward Saliba, Natalie Goleta, and Daniel Banks, who was recently graduated. So this was our gyrotron that was completed about a year ago, and we installed it. Now I'd like to move on to the next topic, which is the short pulse microwave generation from a CW or a pulse gyrotron for application in pulse DNP-NMR. So the point is why we need short pulses. And as most of you know, CW DNP mechanisms scale unfavorably with the magnetic field. So at higher magnetic fields, you need to circumvent this problem, which is done by using pulse DNP techniques, which has gained a lot of interest very recently, and techniques such as novel and frequency selective integrated solid effect and this time optimized DNP. And these techniques require microwave pulses, which have a specific time sequence and which can be modulated in amplitude and phase with time. For example, in novel, here's a pulse sequence for novel, which I took from this paper. So you have a microwave pulse, which is about 16 nanosecond long, followed by another pulse, which is about 100 nanosecond long. And there's a 90-degree phase difference between the X and Y pulse. And there's afterwards a delay of about 40 microseconds. So this specific pulse sequence experiments were done at X band, about 9.7 gigahertz, using a TWT amplifier. And these are the nice results that were published in this paper. Another example is top DNP, where you have a similar pulse length, about 28 nanoseconds, and a delay of about 56 nanoseconds, and the whole thing is repeated after a certain interval. So these pulse sequences are on the order of nanoseconds, tenths of nanoseconds. And nice results were obtained with these pulse sequences using this technique, and they were published in 2019 by Kong. So at higher frequencies, you have to generate these pulses using CW gyrotrons, because you need CW or pulse gyrotrons at higher frequencies. They can put out higher powers. And how do we generate short pulses from these gyrotrons? So one way is to operate the gyrotron using a pulsed power supply. And typically a pulse power supply, you generate a microsecond or a millisecond long pulses. So your microwave pulses are that long from the gyrotron. The other way is to, you can sort of externally modulate the voltage on the gyrotron to modulate the power or the frequency. And that is first reported for DNP gyrotrons in 2015 by Idehara Group. This technique is applied a lot in fusion gyrotrons at megawatt power levels. And so what the way it's done is you bias the body potential by a few kV using an external voltage modulator. And as you know, the frequency is dependent on the voltage. So as you change the voltage, your frequency sweeps. And depending on how fast you sweep your voltage, your frequency would change and your power would also modulate. And you see about this 50 megahertz frequency sort of chirping effect here using this external voltage modulator on the body. And these results were reported in 2015. And later, Barnes group at WashU also did similar experiments where they modulated the body potential by about 2 kV. You can see in this top plot here. And you get this nice power modulation, which is on the order of about 0 to 30 watts. So the pulses that are generated using this technique are about few hundreds of microsecond long, because this is how fast you can sweep your voltage. So in order to generate nanosecond long pulses, as you need in short pulse DNP techniques like novel and ICE and top DNP, you need nanosecond timescale pulses, and you cannot do it using these techniques. So you have to use an external sort of switching method. And this is done using this laser-driven semiconductor switch. So in a switch, you kind of reflect the microwaves from a CW or a long pulse source by inducing temporary reflectance in a semiconductor wafer. So if you have a semiconductor wafer that is in the path of the microwaves, initially the wafer is transparent because its thickness is optimized for the microwave frequency. So initially the wafer is transparent. Then you illuminate your wafer with a laser, and the laser induces a reflectance in the wafer when the laser photon energy is greater than the band gap energy of the semiconductor wafer, and it generates an electron hole plasma in the wafer. When this plasma frequency is more than your microwave frequency, the wafer becomes reflective. And once you turn off your laser, it becomes transparent again. So you can sort of modulate the microwave pulse by turning on and off your laser. And a lot of pioneering work using this technique has been done in the 70s, and its behavior has been well characterized at low power levels. And for NMR, this technique has been applied using a gyrotron at 154 gigahertz by Osaka University and Fuku University groups. And this has also been used on a free electron laser at the University of Santa Barbara at a kilowatt power level for DNP-NMR at about 248 gigahertz. And so we tried to sort of test the laser-driven semiconductor switch behavior over a kilowatt power level, which has never been demonstrated before. So we used our 110 gigahertz gyrotron, which can produce 3 microsecond long pulses at a megawatt power level. We use this gyrotron to test the switch behavior. And so you see a schematic here. There's your primary wafer. So this microwave generated from the gyrotron is fed into the wafer here. So initially it is transparent to the microwaves. Your laser setup is here. So when you turn on the laser, you generate that electron hole plasma in the wafer, which when this plasma frequency is over this microwave frequency, it immediately becomes reflective, and you have your reflected microwave beam coupled through this optics here to your output setup here. When you turn off the laser, it becomes transparent again. So we did these experiments using our Megawatt Gyrotron. You see the picture of the lab. This on the top right here is our Gyrotron. This is the waveguide here. So the microwaves are fed into this waveguide. And this is the switch part here on the left side here. And the laser, it's a 532 green laser, and some laser optics here to illuminate the wafer. And these experiments were done by our grad student, Julian Picard. This is a close-up view of the switch. So this is the input waveguide that feeds the wafer. So this is a primary wafer. It's illuminated by the laser through this mirror here, and the reflected beam comes to the output waveguide here. you can install, you can use the switch using 2 wafers to fine tune or tailor the pulse width of your output pulse. And this is an example of the output pulse we have. So in blue, we have the input pulse. This is about 3 microsecond long total from the gyrotron. And you see the switched output pulse is nice, sharp, about 10 nanosecond long here. And when you look in more detail, so this is the laser pulse here on the right, top right here, which is a green laser at 6 nanosecond pulse length at about 250 millijoules. And the peak intensity at the wafer is a little high, 3.5 megawatt per centimeter square, with that puts out a laser energy density of about 15 millijoule per centimeter square on the wafer. And after illuminating the wafer with this laser pulse, this is the reflected microwave signal you see. Reflected power using a silicon wafer, and the pulse length here is about 9 nanosecond long. So the incident power from the gyrotron pulse was about 525 kilowatt for 3 microsecond long. And using the switching technique, you see a 9 nanosecond long pulse with an output microwave power of about 80% of the input power, about 400 kilowatt. And so we were able to generate these nice short nanosecond time scale pulses using the switching technique from our microsecond long pulse gyrotron. And this can be applied to a CW gyrotron as well. And these results were published in 2019 by Julian Picard. So I'll switch focus now to the pulse phase stability in a gyro amplifier. So we did some experiments to study how stable the phase is in a gyro amplifier for doing pulse DNP-NMR techniques. And because you will need gyro amplifiers at higher frequencies to do pulsed DNP-NMR experiments. Now the point is, why do we measure the phase stability? So pulse DNP requires phase difference between the input and output signals that is stable to within about 10 degrees. It could be lower. This is the number. We know that it's about 10 degrees. And this is an example again of a novel sequence. And so your X and Y pulse, the phase difference here, your Y pulse is 90-degree phase shifted to your X pulse. The point is, when you have multiple pulses repeated one after the other, how stable is the phase within shot to shot and within your one macro pulse? And can it be compensated if you know the phase difference is not really 90 degree? Can it be compensated? So we did some experiments to look at the phase stability of the gyro amplifier, because you cannot directly measure phase at higher frequencies. And so the only ways to look at the phases, you can measure the stability of the phase within the pulse and pulse to pulse. And thus far, there has been no experiment that has done at higher frequencies to study the phase over W band. The gyro amplifier, the W band, there was a study of phase stability. And the existing techniques are very challenging about W band because the limited components, such as the balance mixer, which is the primary component you need to study the phase stability. And this is also important because there are other phase sensitive applications which require an accurate phase measurement on your device, like the coherent radar and communication systems, accelerators, and linear colliders. So studying phase stability in a gyro amplifier is very important to not just the DNP, but to the other fields as well. So we used our 140 gigahertz gyro amplifier to study the phase. The gyro amplifier was built and completed some number of years ago. and it operates in 2 microsecond long pulses. We got a very nice device performance, 20 dB gain, and a nice circuit gain of about 30 dB from this amplifier, and a saturated output power of about half a kilowatt. So we sort of optimized the operation of the amplifier to study the phase, because phase measurements are extremely sensitive. So we chose a voltage range where the gyrotron was the peak performance more stable, and the beam current was about 1 amp. We did not really operate at the nominal beam current. And the input driver we used was a 2-watt extended interaction oscillator from CPI EIO. And the output power for these measurements was about 30 watts. So the amplifier performance was optimized for these measurements. So the measurements were done using a balance mixer. So in a balance mixer, as you can see on the picture here, you have an RF and LO ports. And if your RF and LO frequency are same, your intermediate frequency that is coming out of the balance mixer is a DC voltage if your RF and LO frequency are same. And then your DC level is a function of the phase difference between your input and output signals. So if your frequencies are same, if your phase for both signals are same, your intermediate frequency is 0 and your VDC is 0. And when you change the phase in one of the arms here, your DC voltage level becomes a function of the phase. And that way, if one signal is coming from the amplifier and the other signal is your reference signal, you can control your reference signal. And by introducing an external phase into the reference signal, you can do a mismatch of the phase here in the mixer, and you can record your voltage as a function of the phase, where you can sort of deduct the phase performance of the signal that's coming from the amplifier. So we did that. So this is the driver here. So we split the driver in 2 signals, one that feeds the amplifier, and the amplifier output is fed into the RF of the balance mixer. The other signal is fed into the LO through a variable attenuator and a phase shifter, where you can introduce the external phase into the signal, and you can record your DC voltage level. And the reason why we did these experiments at a 140 gigahertz amplifier is that because balance mixers are not available above 170 gigahertz. So you have to do it below 170 gigahertz. So this is the diagnostic setup here in the CAD version. So we have our EIO driver here that feeds the amplifier with, but before that, the signal is splitted here using a beam splitter that goes into the other arm through an attenuator and a phase shifter, and then it goes to the mixer here. The other arm goes to the amplifier. It gets amplified. The output of the amplifier is fed into the RF port of the mixer through this attenuator, and there are some bunch of monitor diodes for measuring the response at every voltage before we feed into the mixer here. So the system, the measurements were extremely sensitive to small changes in the phase or the RF power level we are feeding into the mixer on both RF and the LO ports. So we did a thorough calibration at every voltage we did the measurements. And you see the experimental setup here. This is Dr. Rosenzweig. He did these experiments. And this is the waveguide that comes from the driver, feeds the amplifier, it gets splitted before here and goes to the one arm of the phase, the balance mixer somewhere here. And these are the results we got. So you see, as a function of voltage, the measured phase, and a nice d phi over dV of about 130 degrees per kV was measured. And we also did use Gyrotron code to simulate the phase. And we got a nice, excellent agreement between our experiment and simulation. So 120 volts per kV is the simulations, and the measured is 130 kV, 130 degrees per kilovolt. And the phase difference was extremely stable as long as the operating conditions like the voltage, the beam current, magnetic field, everything was maintained. And if you do a quick math, so this for the phase stability as per the voltage level, so for achieving plus minus 5 degrees phase stability, a voltage stability of 40 volts is needed in this range. So we are operating at about 46 to 48 kV. So at 40 kV, this is about 0.1% stability, which is required from the power supply, which you can get. I mean, it's hard, but you can get it. So phase, it's found to be within the agreement for the requirements for DNP, and these experiments were completed and reported and published earlier this year in infrared millimeter waves. So in summary, we have completed our Gyrotron, the 527 gigahertz Gyrotron, and installed with the 800 megahertz DNP spectrometer. The Gyrotron has about 9 watts stable output power and about 400 megahertz of frequency tunability. Our switch, we were able to generate up to 9 nanosecond long pulses at 110 gigahertz using our Megawatt Gyrotron using the laser driven switch. And the phase measurements at 140 gigahertz were the first time ever reported. And the experimentally measured phase difference on the cathode voltage was 130 degrees per kilovolt. And this phase stability, which is found to be less than 10 degrees, meets the pulsed DNP requirements. And I would like to acknowledge our group, Rick Temkin's group at MIT, and Bob's group, and the funding agencies, NIH and NBIB. And thank you all for listening to the talk.
All right, thank you very much, Sudheer, for your very interesting talk, and also like to showing us the prospect of DNP and possibly pulse DNP happening at Highfield in this summer, or maybe beginning next year, as much as earlier as Bob would like to be. But anyway, before we go through the question in the Q&A, maybe I will ask like a few questions first, so that the participants have time to type their questions into the Q&A. So I'm just curious because, like, you know, of course, I'm familiar with some of the things that you have described, but I'm just curious because for the 140 gyro amplifier, you are using the TWT technology, and then for the 250, you use the photonic bandgap PBG. So, like, if you were to build one at even higher frequency, let's say for 527 or possibly 792, which technology would you pursue, and what is the advantage of one over the other one?
So at higher frequencies, I think photonic bandgap circuits like the one we have in our 250 gigahertz Gyrotron would be more advantageous because if you look at the results that were published from the 140 gigahertz and the 250 gigahertz, at 250 we got about 55 dB of circuit gain from the photonic bandgap, and the overall device gain was about 40 dB. In the 140 gigahertz Gyrotron, which is a confocal waveguide circuit, we got up to 24 dB device gain and about 35 dB of the circuit gain. So clearly, I mean, you see more gain from the photonic bandgap circuit. Another advantage is photonic bandgap circuit is a highly frequency selective, and by tuning the lattice structure, you can make the operation of the amplifier really in one mode and not the other modes not affecting the amplifier operation. So the photonic band would be go higher frequencies.
When you go to higher frequency, I mean, how much would you, I don't know, like, what is your expectation of the machining precision on the lattice structure? Because I can imagine it would be more difficult to machine this
So it's not just the machining, it's also the driver power. Like you don't have enough driver power at higher frequencies. At 250 gigahertz, I mean, we have now these VDI sources, which we call like high power VDI sources at 200 milliwatts or to maybe to 300 milliwatts. If you go to 460 or 527 gigahertz, your driver power is like 1 or 2 milliwatts. Mm-hmm. And so the total output you get from your amplifier would be like 1 or 2 watts after the amplification. So going at higher frequency is still in the long run, but about 200, 300 gigahertz is there. You can make your amplifiers there and propose DNP. Mm-hmm. In terms of precision for manufacturing these photonic bandgap circuits, within like 10, 15 microns, which is easily available. You can do it in direct machining. You can build your circuit with that, and you see your performance. What do you expect, like within your desired numbers? So it's not super tight on the tolerances at these frequencies. Obviously, if you get a higher frequency, it has to be really, really precise.
Mm-hmm. And of course, like, yeah, my second question is that when you measure the phase stability on the 140, of course, you verify it using the Maggie simulation. So I was wondering, of course, you mentioned that you don't have a balance mixer at 250, but of course you can also do the Maggie simulation on 250. So would you expect to have the same behavior, the delta phi over the voltage?
Yes, you can do those simulations at higher frequencies, and I think the phase, I'm not sure how it varies with the frequency, but it would be probably about the same order, plus minus 5 degrees per volt.
Okay.
So yeah.
Okay. So I will go to there's only one question now in the Q&A. You can open it up, but I'll read it out. So Chukun Gao, I assume from ETH, she asked, would it be easier to fabricate a fundamental mood gyrotron within a high field magnet than a second harmonic one within half the field?
Okay. It's a balance. Yes, you can make a fundamental mode gyrotron. It is difficult in terms of fabricating it, but yes, you can make it. The question is your cost estimate. If you make a fundamental mode gyrotron at 700-800 gigahertz, your magnet would be 20-22 Tesla, which becomes very expensive. But in theory, yes, you can do it.
Yeah. And if I'm not mistaken, if you make a second harmonic one, the maximum theoretical efficiency is only 1/n squared, which is 25% of the fundamental one.
Yes, operating its higher harmonics, your frequency goes down. At higher frequencies, if you look at some of the results from these DNP gyrotrons, the power requirements, the output power is about like 50 or 100 watts. Mm-hmm. And you don't really care about the efficiency at these power levels. I mean, gyrotron can easily produce hundreds of watts of power. Mm-hmm. But operating at higher harmonic efficiency goes down, and typical operating efficiency of experimental efficiency of a second harmonic gyrotron is about few percent. I mean, in theory, it's about 20%, but in reality, it's about a few percent.
And do you worry more about hitting other modes if you work in the second harmonics?
Second harmonic gyrotrons are, yes, they're extremely difficult to operate because it's super sensitive to the alignments. And you have to carefully design your operating mode for the gyrotron so that it's far from the other fundamental modes.
Yeah.
Yeah, it's a balance. You can make one or the other, but it's a balance, yes.
Mm-hmm. And then the next question from Beyond Costinius from Rostock. What is the intrinsic limit from the excitonic plasma on the pulse 4 times?
Yes. So with in our switch using a laser, the silicon wafer, the pulse length was about 9 nanoseconds. If you use a different wafer like gallium arsenide, your pulse length is about 24 nanoseconds. So there is a difference, of course, and that comes from the limit on this excitonic plasma, as Bjorn wrote it down on the pulse hold time. And but you can generate even shorter pulses by combining the two wafers together. You can even go down in up to 3 nanoseconds long pulses.
Is this somehow, is this even, or maybe should I ask, can you, what about the refresh rate or repetition rate that you can do this?
Using these semiconductor wafers are not good thermal conductors, and you're depositing a lot of laser energy onto the wafer that gets absorbed onto a very a thin layer on the surface of the wafer, about a few microns. And the rep rate we run at, it's about 5 hertz max. If you want to go at higher rep rate, it's very difficult with the wafer. You would have to come up with some other technique because the wafer would break.
Yeah, 5 hertz. Yeah. Okay. Yeah. One every 200 milliseconds. Okay. Yeah. Okay. So the next question from Blake Wilson. What is the shortest pulse length that can be generated using the optical pulse slicer? Do you see the carrier recombination time vary with the microwave power?
Yes. So the shortest we generated with a single wafer was 9 nanosecond. Using 2 wafers, we generated 3 nanosecond long pulse. And what's the next one?
Do you see the carrier recombination time vary with the microwave power?
The carrier recombination time, I don't know, I have to look. There was a.
Can you increase the rep rate with? Can you increase the rep rate with using lower microwave power?
You can increase the rep rate with the. So the microwave power does not get absorbed into the wafer. It either gets transmitted or most of it reflected. So the microwave power does not really have much effect on the wafer. It's the laser energy. So if you can switch the wafer with faster laser, high rep rate laser, your microwaves will get reflected on the same time scale. Mm-hmm. But the carrier recombination time, I think slightly depends on the microwave power, yes.
Okay. Bob, do you have other questions or other advices or before you or I introduce the next speaker?
Well, one comment is that you were talking about the power available at say 527 from diode sources and things like that. So in a conversation I had with VDI, they said they were trying. You know, they've done very well at 250, 263 gigahertz. They can generate about 200 milliwatts there. And they're trying a similar strategy now at 395, which is for 600 megahertz. And they're going to try it at 527, where it's a brute force strategy, basically, to put more diodes in parallel and, you know, to recombine them to generate more power. Now, how successful that's going to be at generating enough power to drive an amplifier, you know, who knows? But that is one approach that they're pursuing at the moment. So that's and there was actually another question that popped up here from Martha Millen. She says, "What about the longest pulse period or wafer of wafer reflection? Was this limited by the laser only or just the wafer?" Can you see the? Yeah.
It was limited by the laser. I can show 1 or 2 slides. I don't have a backup slide in this one here. Just give me a second. have to stop share here and show one slide, just one slide from this one here. So this is the top one is the laser pulse, which is about 6 nanosecond long. And you see these 3 different cases where we have the silicon wafer, which is about 9 nanosecond long, and the gallium arsenide wafer, which is about 24 nanosecond long, and this dual wafer, which is 3 nanosecond, combining 2 silicon wafers together. And after the laser pulse stops, the silicon, the pulse we see with the silicon wafer, it kind of stops here, but this gallium arsenide wafer kind of goes a little longer and then comes down. So the recombination takes a little longer in gallium arsenide wafer compared to the silicon wafer. So depending on which one you use, you can generate, like we were able to generate up to 3 nanosecond long pulses using 2 wafers.
Okay, all right. Well, we should probably go on to the next.
We have one more question.
Another question, yeah. Okay, how expensive will such an amplifier be compared to a 9 Tesla Gyrotron? I can't afford one, but just curious. It's an anonymous question.
I don't know. I mean, the gyro amplifiers for DNP are not sort of commercially available. You have to build one your own, and compared to a Gyrotron, it's probably about, I mean, the primary cost is the magnet and the power supply, of course, the second one. So it's probably 2 times, maybe.
Well, the CPI people have said they can do it for, maybe 10 or 20% more. That was their, when I've asked them if they would do it commercially. So who knows though, yeah.
Yeah, I mean, they foresee sort of like more units being developed in the future so they can bring the price down.
Yeah. So it will be somewhat more expensive and somewhat is yet to be defined.
Yes.
Okay.
Yeah.
Okay, so let's thank you very, very much, Sudheer, for a very interesting talk. And let's go on to the second presentation, which is from Kendra Frederick. And Kendra was an undergraduate at the University of Michigan, where she majored in biochemistry as well as French. And she got a master's degree in Paris. So she's become a real Francophile. And her PhD with Josh Wand at the University of Pennsylvania. And she was then a postdoc at the Whitehead Institute here at MIT with Sue Lindquist. And now she's an assistant professor at UT Southwest Med School in Dallas. And she's going to be telling us about in-cell structural biology using DNP MAS NMR. So Kendra.
Thank you, Bob. Right. So let me get this going. So yeah, this is very much about what you can do with a constant wave and much more applications, although really excited about the possibilities for pulse DNP, and we'll have applications ready when we get there. So what I want to talk to you about today is really manifest in this picture, and every single cell on this plate is identical genetically, but they have really different phenotypes. And the difference between them is purely the conformation of the single protein. So the phenotype I'm showing you here is colony color, and it's very beautiful. But if I were to plate these cells on a different plate, it would be the choice between life and death. And so my lab is really interested in understanding how protein conformation can affect cellular fate. And this is manifest, of course, with many neurodegenerative diseases where there's proteins that have one conformation during a person's lifetime, and they take on a different conformation which is associated with toxicity. And the problem with these diseases really is that they're protein folding, they take it on a different conformation, and there's not a single therapeutic strategy that gets at the underlying protein pathology. And possibly this is because we're not looking at the right conformations. So for many proteins, they have a a simple folding pathway, and they end up in the energy minimum and have this native conformation here. And we can study that biophysically, and the conformation we get in the test tube is the same as the conformation in the cell, and we can verify that with all sorts of secondary assays, like an enzyme assay. You get the same activity in the test tube as a cell. But if the environment is more complicated, or if the protein is metastable, you can end up in a variety of different conformations. And clearly the environment can change the arrows and change where you end up conformationally, right? And so inside of the cell is really complicated. There's lots of different things that can interact with your proteins. And so particularly for these proteins that can take on more than one conformation, we want to be able to study that protein conformation while everything else that interacts with it is there. Right. And so NMR is really uniquely suited for these types of studies because we can get atomic level information simply by putting our isotopes on the protein that we want to see and depleting isotopes from everything else, but leaving it there. Okay. And it's actually really important. So the protein that I showed in the beginning is the yeast prion protein. And so you can take on a prion conformation or a normal conformation. And this prion conformation, it can take these normal conformations and self-template into a second conformation that has a different biological activity. And then inside the cell, it needs to interact with chaperone protein to break up these prion conformations, and so they could be passed on to daughter cells, right? And so for yeast, HSP104, the chaperone protein is involved in this process. And having that molecule around is important for the activity. And actually having the stoichiometry correct is also important because, if, for example, if you were to knock this protein out, you don't get inheritance anymore because HSP104 doesn't interact with this conformation. And so you lose inheritance. And if you overexpress this protein, it works really, really well. And it destroys the prion conformation. You no longer get inheritance. And it works in concert with a variety of other chaperones which have known influences on the inheritance of this protein, or the inheritance of the prion conformation, right? And so all this to say is that for these proteins that can take on multiple conformations, the identity of the molecules in the solution, the stoichiometry, all of that is important. The concentrations are important. And so if we are to make a sample where we have our yeast prion protein, for example, isotopically labeled in a background that we cannot see, and we compare that sample to that same protein in the prion conformation in isolation, we can see that the biological context does indeed influence the structure, right? So it's important to include it when looking at proteins that can take on more than one conformation. And in this particular case, actually, it's these regions of intrinsic disorder off of the central conformation right here, that's the amyloid conformation, that take on a different conformation inside of the cell, or inside of lysates. Okay. So we really want to do in vivo structural biology because we want to maintain the biological integrity of the environment of the protein that we're studying. Right? And so the way that we get specificity is to think about isotopic labeling and also using homo and heteronuclear pulse sequences to select for adjacent atoms. Right? We get our sensitivity in order to detect proteins at their endogenous concentrations by using DNP-NMR. And then I'll touch a little bit on this at the end. We get resolution through, or at least in my group, we get it through protein chemistry by using specific and segmental isotopic labeling, although I won't talk about that very much today. Right. And so in the case of neurodegenerative diseases, I'm going to mostly talk about Parkinson's disease today, or alpha-synucleinopathies. So the protein that takes on a different conformation inside the brains of people afflicted with Parkinson's disease or other alpha-synucleinopathies is alpha-synuclein. Okay. And these aggregates can take on not one, but a variety of molecular structures. So all of these different conformations that were shown here, so you can see the variety of different beta sheets that are spread out throughout the primary sequence of alpha-synuclein. They're all in different places, right? These are all in purified settings. And in fact, these structures right here are cryo-EM structures of brain-derived fibrils. And so in they studied 5 different individuals that had different mixtures of these conformations right here. And so alphanucleon not only takes on a variety of fibril conformations, it also can take on these disordered oligomeric conformations, perhaps these helical oligomers that were once thought to be the toxic structure. It can associate with membranes and take on alpha helical conformations. And there's a really beautiful saturating mutagenesis study that came out recently that suggests that this might indeed be the toxic structure. And it's present in solution as an intrinsically disordered protein. So it's definitely a protein that is very environmentally sensitive, right? And so the problem with all of those previous studies is that it's always a purified molecule. You have eliminated the environment in order to figure out what the structure is. And so my lab's goal is to figure out what the conformation is inside of cells. Okay. And it's really particularly important for neurodegenerative diseases because the field is fraught, and that, you know, you get a conformation and then everyone's like, oh, but you altered the system. And so you don't actually know what's involved with toxicity. So the goal here is to get cells that have a phenotype, do a structural experiment, and then test afterwards. And so if you have the same phenotype before and after the experiment, then you know that the conformation or the conformational ensemble that you've determined is reporting on something real. And so in this context, if you then alter the phenotype by adding a drug or a genetic modulator, and you get different toxicity, you can then correlate toxicity with structural ensemble and start to understand what the causative toxic conformation might be in these diseases. All right. So the trouble is like, we want to sort of bridge the gap between the physical chemistry or these NMR experiments and a biological system, right? And so for DNP, you have to have millimolar concentrations of a polarization agent, which is a stable radical. You spin at the magic angle for thousands of hertz, and you have radio and microwaves in a magnetic field, right? Although the idea actually is really simple. So you grow cells that have whatever setup that you want. You add the polarization agent and a cryoprotectant, right? You freeze the cells and you put them in the spectrometer, and at the end you just thaw them and you plate them and you test them again. Okay. So first things first, cells tolerate millimolar concentrations of the polarization agent Amupol. And so what I'm showing you here is membrane integrity over time with a variety of doses, right? So we haven't ruined everything just by adding the necessary agent for DNP. The other thing, which is perhaps not a surprise for biologists, but might surprise some physicists, is that mammalian cells do not survive flash freezing. You drop it in liquid nitrogen, at the end, you're not going to end up with anything that's alive. So if you are a spectroscopist and you're putting your sample inside of the magnet, if you're going from room temperature to cryo temperatures, 100 Kelvin, where we usually do experiments, basically you've just sliced everything in that sample before you start the experiment. But if you slowly freeze them, if you reduce the temperature at 1 degree Celsius a minute, everything's fine. And so all you have to do is figure out how to transfer a frozen sample into the magnet. And that's what a team of scientists in my group has done. Okay. So that part is set up. I want to spend a little bit of time talking about how we're sure that the sample is okay. Okay. And to do that, what we did is tested cellular viability via a variety of methods at all these different points in our preparation protocol. Okay. And what do I want to say about this? Right. So at first, I'm going to talk about this trypan dye exclusion. It's a common assay, and it basically is just looking at membrane integrity. You either exclude the dye or you don't. Okay. And so if you take cells right off of plates, you have really high viabilities. And then if you add DNP juice, in this case, we're using 15% glycerol, we're deuterating, we're adding PBS instead of any sort of FBS or other sort of media that people use. And this is purely because we don't want any confusion of where all of our signals are coming from. I don't think that this is going to be an issue. If you need to use FBS to keep your cells alive, it should be fine. But we tried to eliminate carbon so that if we're looking at really low concentrations of a protein, we don't have any confusion from the background. The addition of glycerol slightly reduces the membrane integrity of these cells. This is known from the literature, but it doesn't, this is a standard procedure. If you freeze them slowly, there's not a statistical difference between these two populations, so slow freezing has no additional effect on viability. And if you pull the cells out of the magnet after, you have a 15% loss, right? So lots of things have happened there. You have magic angle spinning, you have the transfer into the magnet, but you also actually have taking off of this end cap and the silicon plug, right? And if we look at paired experiments, we find that we have less loss in viability. So if we look at the same sample here as here, the ones that have high viability here have high viability there. So sample handling matters. And so if we look to see if there's any difference between samples that have been frozen but not measured by DNP, not subjected to MAS, there's absolutely no difference between them. So once a frozen sample is inside of the spectrometer, there's no further damage, right? So these cryopreserved cells right here are most representative of the measured sample. Okay. We did two other sets of controls, right? And so we did a cell growth assay. Basically, after we just grow the cells on plates and measure their density over time, their confluency over time. So if you add the glycerol, again, this is known, it delays the growth, but it doesn't change the growth rate, right? And then neither freezing nor the NMR changes the growth rate if you look at something that's been frozen and then removed from the rotor. So mammalian cells are not only viable, but able to propagate. And in fact, this change in membrane integrity doesn't necessarily correlate to the ability to propagate. Okay, I guess I should also say deuteration and amupole don't affect the ability to propagate either. Right. And finally, you can check out your cells by microscopy. I'm showing you images from these two time points here, but you could look at the end as well. And so you can use fluorescence microscopy to verify your samples. Okay. So there we go. We can we have a system where we can check cell integrity before and after the experiment. Second problem is that we need to get amupole into the sample because amupole, in order to really enhance the protein that we want to see, it requires proximity. Right? And so the question is, can amupole polarize these cells? Because they're surrounded by a plasma membrane, which is usually pretty good at keeping things out. This is a 750 Dalton small molecule. Right? And so in order to figure out if we're getting the amupole in or not, we first figured out if we could use, we first established biological markers that we could use that are distributed throughout the protein. So lipids are at 35 ppm, proteins have this moiety right here, RNA and DNA there, right? And so if we integrate the area underneath these peaks here, we find that we really recapitulate the biomass percentages that we expect from that are determined by chemical means, right? And so now we can ask, can amupole polarize the cells? Because now we have sort of a spatial readout through chemical shift, right? And the answer is yes. DNP enhances all the biomass components in these intact cells, and it does so in a concentration-dependent manner. Moreover, we know that amupole enters the nucleus of the cell, so there's no barrier once it gets inside to getting inside of the nucleus. And we can tell because RNA and DNA, of course, have different chemical shifts, and we can see strong signals from DNA as well as RNA. Okay. So here we are. We now have a system and a protocol to figure out or to be able to measure viable frozen samples that can be enhanced well by DNP so that we can get the sensitivity we need to see proteins at low concentrations. And so what we do is we take isotopically labeled protein that's been prepared recombinantly, and we introduce it into the cells by electroporation using the methodologies that are really perfected by Phil Selinko's group a while ago. Okay. And so I'm going to talk about alpha-synuclein. And we really want to understand which conformations are sampled in health and in disease for alpha-synuclein in these complicated settings. Right? So before I launch into what that answer might be, I need to establish that DNP usually gives broad peaks, but nonetheless, for alpha-synuclein, we are able to distinguish between a variety of different conformations of purified samples under DNP conditions. So we have these helical conformations that are on nanodiscs, frozen, intrinsically disordered alpha-synuclein, and then two different fibril forms. This one described by the Reinster group a while ago, and then one with an extended amyloid core. And so the different structural elements are plotted here against the primary structure or the primary sequence, right? And so I'm just going to look at distinct regions. So for example, we can look at the glycines right here, or we can look at threonines right there. And using DNP, we can distinguish between alpha helical conformations, this is for the glycine region, and these amyloid fibril conformations. You can see a strong peak for the beta, and then sort of intermediate values, which would correspond to these intrinsically disordered regions right here. Okay. And if we look at the threonines, we see the same thing. And in fact, this is repeatable. So this is two different preparations of this fibril type right here with slightly different labeling patterns, although always the threonine is labeled. And we even recapitulate this hump. Okay. So now we introduce the labeled alpha-synuclein into cells, right? By electroporation, take it through the same protocol. If we put a fluorophore on the alpha-synuclein and electroporate it in, you can see that the cell membrane is in orange and the fluorophore is red, and this protein is diffused throughout the cell. And we can collect spectra with about 24 hours of signal averaging on these samples, which probably have somewhere between 20 and 30 micromolar alpha-synuclein. And these are carbon-carbon correlations. So if we look at this region that corresponds to the backbone, you can see that alpha-synuclein inside cells seems to be a mixture of alpha helices and random coil. All right, so that's the black spectra right here. But if we look at specific amino acid regions, we can see that actually surprisingly, while you do have some random coil conformations, we have really strong alpha helical signals. So this is a little bit of a surprise because many people are probably familiar with the work of Phil Salenko's group, where alpha-synuclein inside of cells by using solution state NMR spectroscopy was described as a compact intrinsically disordered protein. And the quantification of these experiments was actually quite good. They used low micromolar, 10 to 20 micromolar concentrations, and didn't seem like they were missing a whole lot of signal, right? However, what we see right here is a combination of strong alpha helical signals with some intrinsically disordered regions. But this is frozen solution, this is frozen, and so it should report on the entire structural ensemble. So I want to come back to this question about can amupol polarize the cells, right? So we know for sure that amupol enters the nucleus, right? Because we see these strong signals for DNA, right? But one thing that I didn't say earlier is that if you take cell lysates rather than intact cells, we can get really, really high enhancements at really, really low concentrations. This is 5 millimolar right here. But we don't get nearly that enhancement for the intact cells. And this is the buildup time right here. So we're getting really big buildup. So this goes up and then goes down, right? And if we look at the buildup time, if we look at the primary data, if you look at just a sample of proline, a standard sample of proline, this fits really well to a single function. You get a really small residual. And then if you look at this sample with the lysed cells, so there should be no barrier because the cells are completely lysed. The amupol has access to everything, right? You expect a slightly broader thing because you have different chemical environments for the different carbons, but overall you have a really small residual. But when you incubate cells with the amupol, you have a really big residual behind. So this doesn't fit well. It's not described well by a single buildup time. And that's probably related to the fact that you don't have a homogeneous concentration of amupole throughout the measured biomass. So this implies that the observed conformation is going to have some sort of spatial bias, right? So the idea then becomes, well, why don't we just bypass the cell membrane and transiently permeabilize the membrane to introduce the amupole in by electroporation, sort of in the same way that we did for the alpha-synuclein in the first place, right? And so we did a whole bunch of controls to make sure that we still have cellular viability after this process. And then when we look at the residuals for electroporated cells compared to these other things, you can see that the residual, you were very well described, or much better described by a single function than you are for the intact cells right here. So the amupole concentration is homogeneous and electroporated into cells, right? Moreover, if you look at the relative concentrations of RNA and DNA for these different preparations, and then compare it also to the other components that you see in the cell, the intensity of the protein peak or the free amino acids, right? You can see that the peak intensity when you incubate for DNA is much smaller than you expect for the RNA or for these other components, right? So this suggests that there's an amupole gradient in the incubated cells. So when we electroporate, the amupole and the protein in at the same time, what we see is now no longer this really strong alpha helical signal, but rather a much broader signal that's commiserate with what you would expect for an intrinsically disordered protein. So here's alpha helix, here's beta sheet, and sort of here's what's in between. So this suggests that something that hasn't seen before, and that is alpha-synuclein inside cells has at least two distinct conformations. One is strongly alpha helical, and the other has a much broader distribution. And then if you look at the comparison between the lysed sample and the electroporated sample, there's a suggestion, and we're still working on isolating specific amino acid regions for this because it's a pretty messy spectra, but the sample has a reduced conformational space in electroporated cells relative to lysis, right? And this is also manifest in the carbonyl region. When you looked at the lysed cells, you have a broader peak than you do when it's electroporated. Right? And so this is actually really nice because then it's recapitulating the solution state NMR result, which suggested that inside of cells, alpha-synuclein is a compact intrinsically disordered protein. But in the lysed environment, you would expect that it could sample a broader amount of conformational space, right? So if you're thinking about in-cell structural biology, I guess the reason that we do it is because we want to have the identity of all the interactors maintained, the stoichiometry maintained, their concentrations also is important, that's maintained. This is an overexpression system, and the organization also maintained. And so in incubated samples, solution state samples, electroporated samples, there is that transient permeabilization. You could potentially lose something. This is all intact. If you lyse the cells, you lose that tight concentration because you're diluting into the buffer, and you lose the organization. But of course, every experiment has a bias. And so in solution state, you don't have a spatial bias, but you're going to have a bias for dynamics. So anything that interacts with the membrane or makes a large complex, you're not going to see anymore. With incubation, it seems like we have a spatial bias because the amupole is not penetrating all the way into the cell with the same concentration. When you electroporate, you remove the spatial bias, right? But for DNP, because the sample is frozen, we no longer have a dynamic bias. Everything is rigid. Okay. So now we have a situation where the in-cell structural ensembles are similar for the solution state and the solid state. You have a compact IDP. And then we get a bonus out of this because we can isolate an alpha helical subpopulation. So alpha-synuclein is sensitive to changes in cellular localization, crowding, and local organization. And that's something that we can assess using this methodology. Okay. So I'm finishing up, but I do want to just mention some work that we're doing to work on, well, this is not really resolution, but chemical shift degeneracy. And so, of course, you can not label this is a yeast prion protein, but not label all of the amino acids in the protein, but only label a small number of them. And when you do so, you go from a situation where you have a really broad peak to a couple individual peaks because you've only labeled a small number of amino acids. We're doing that for alpha-synuclein right now. And hopefully next time I talk to you, I can tell you about our work with a segmentally isotopically labeled version of it. Okay, so a quick overview of where we're headed. One of the reasons we chose this cell line, this HEC293 cell line, is because it works as a sensor cell line. And so if you expose these cells that overexpress alpha-synuclein attached to a GFP molecule, and that stays soluble, but if you expose them to fibril versions of alpha-synuclein, but not fibrils of other proteins, they form aggregates in response to that stimulus. Right. So the idea then is that we can add isotopically labeled alpha-synuclein to cells, these sensor cell lines that harbor aggregates, and get information about aggregated forms inside the cell. Right. And so, you know, the cells would harbor an induced prion conformation, and we add our isotopically labeled protein. And it has all of the components it needs inside. Right. And so when we do the control of this experiment, this is work done by Yaka. Here is the aggregate inside of the cell. And if you add alpha-synuclein exogenously, it co-localizes, right? So you get this templated conformational conversion. And so now we can figure out how healthy and diseased biological environments influence the protein conformation, right? Because we can take these cells, expose them to different fibril conformations, and then see what happens, right? So we can figure out which fibril structures are propagated inside cells, and we can also look at regions of intrinsic disorder to see if they become ordered in these biological samples like they do for the yeast prion protein, right? The other thing that we can address is, you know, I spoke about how this multiple systems atrophy had multiple different conformations that were associated with MSA that were fibrils that were derived from patient brains. Right? We can take brain lysates and put them on the cells as well and template these conformations. And so then we can figure out which of these conformations are templated inside of biological environments. And we can also figure out if it's only one or rather a mixture, because all those different conformations were present in the same individual. Okay. And so now I'm going to end, but I do want to highlight most of this work today was done by two different postdocs, Yiling and Yaka, and a graduate student, Rupam. And also say that if you're interested in biological systems and DNP-NMR, we're definitely looking for scientists to help us out.
Okay, thank you for a very interesting talk. You know, you're worried about getting Amupol into the cell, right?
Yeah.
I mean, I guess you know, quite some time ago, you know, we, and looking at your favorite little peptide, GNQQNY, you know, we showed that you could get spin diffusion over 1,000, 2,000 angstroms. Now, that's not quite the cell dimension, but that was only the size, that was the size of the crystal. And it would probably diffuse farther, you know, had the crystals maybe been larger, but those were the size of the crystals of GN. Now that you have to qualify that in that there was really nothing moving around in that particular crystal, whereas you probably have some methyl group hopping still occurring at various places. But I would think it would be very hard to differentiate between just spin diffusion, and the fact that whether or not Amupol, I mean, do you have a fluorescent probe you could attach to Amupol and see it going inside the cell?
Yeah, it was in the moment you
it
do
is.
that, you end up with a different molecule with different properties because it is so small.
Right.
So one thing that I really want to do is get someone with an EPR microscope to take a look.
With a what?
EPR microscope.
Well, you don't have
we had some resolution issues with that, but we're trying. We're trying.
Right.
It's a slow project. I mean, I think that, I mean, the bigger problem is that, like, if you have these concentration gradients, you have regions where you have too much and you get bleaching, right? And then, this diffusion in. So if it's not well mixed, then I think that everything you see is going to be true, but you can't take it as a quantitative measure. So all the structures that you see are there, but you don't know about their relative proportions.
Mm-hmm. Not necessarily, yeah.
Not necessarily, yeah.
Yeah. Mm-hmm.
So I think that attaching the radical directly to the protein is really the way to go, because then you eliminate these questions about dispersion. But aside from altering the molecule, right now at least, we reduce the Amupol on a time scale of an hour or so, and so it's not a viable solution.
Yeah. So there's a question from Gil in Israel. Do you know if the helical synuclein form is anchored to a membrane, perhaps from the outside?
So I would assume that it's probably membrane associated, and it seems likely that it's associated with the plasma membrane, just given the fact that it seems like we have a gradient from high concentration outside of the cell and then lower concentration in the nucleus. But I don't have a good way to assess that. Not yet.
Mm-hmm. Yeah. So you were also worried about resolution, right?
Mm.
And my default option is to tell everybody to spin at 40 kilohertz and at 800 or higher. But have you tried, say, 1-3 glycerol labeling or 2 glycerol labeling experiments to see if how much that improves your? I mean,
yeah, the issue that we run into is one of selectivity. So we're really leaning heavy on proximity of the two labeled sites to distinguish the protein from the background, because just by like a numbers game, there's more carbon from natural abundance in the sample than there is from the protein that you want to see. So in order to highlight it, you really need the proximity. If you separate out the carbons, then you just increase the background as well. So it becomes harder to distinguish between background and signal. So we haven't done that yet. I think that the first thing we're going to do is a lot more specific and segmental labeling. I don't think that our biggest enemy is the line broadening from like dipolar truncation or coupling or relaxation. I think that the biggest problem is there's just too many atoms right now.
Well, yeah, okay. So we can come back to that maybe. Now, Gil has another question: is do you have a way to measure PC decor? I'm not sure what PC decor is.
Oh, we have not looked at variations in the decoration of the lipids at all. No, we don't have a way to do that. I mean, there's lots of things you could think about doing, but no, I haven't looked at any sort of specific modification. It's something you could think about though. It's hard because like a lot of that maybe would be carbohydrate driven, and we do have a lot of signals that are sort of in that same region from the RNA and the DNA.
Okay, Christian Griesinger has a question. You mentioned that you had beta strand resonances in addition to the alpha helix. did you try to see whether this changed by knocking down the HSPs, the heat shock, such as Hiller-Reek did? If this was early aggregates that you see, you might increase their population by that experiment. Or where do you think that these beta-strand alpha-synucleins reside? And did you try thio-S staining afterwards? So there are a bunch of questions there.
There's a bunch of questions. Yeah, you're going too fast. You're going too fast. So I think that there's a lot of ground to cover there. Right now, I think that my biggest goal is to get away from a toy system and into a system where we're actually seeing biological activity, or like there's some sort of actual real biological problem to solve, because alpha-synuclein in a HEC-293 cell is cute, but it's not going to get at answering a question about toxicity. So I think that, you know, for the in cell lysates, we see beta helical character that's more sampled than we do for intact cells. And that's probably just sampling. You get a wide ensemble there. No, we haven't done any of the thio-S controls, but right now there's no indication that in the HEC-293 cells that, you know, there's any sort of stable structure. I mean, unless we induce it. So just soluble, there's no indication of aggregates.
Christian, does that answer all your questions? I'm not sure. You had several concatenated questions here. You can, yeah.
Yeah. Can you hear me now?
Yes,
Yeah.
we can hear you, yeah.
Yeah, well, I mean, more or less, but you just said you don't see beta strand. Then maybe I misunderstood from your from your talk. I thought you see alpha and beta, but so this is not what you're saying.
So, I mean, what we see, inside of the cells is really a, you know, when we see a really broad peak, and that's what you would really expect for a frozen intrinsically disordered protein, right? And when we lyse it, we get a larger, so this beta right here, this is from a purified sample of bona fide amyloid fibrils, to just give a reference of what we see for that. So I don't think, if there are stable beta conformations in this toy system right here, where we electroporate alpha-synuclein into a HEC-293 cell, it would be a small amount. And, you know, we haven't investigated that, we haven't done like FRAP or anything like that, but it's not something that we see any evidence for. Like there's no speckles in these, for example. So if we go to this system where we've induced amyloid conformations in these sensor cell lines and then add the protein to it, we still are dealing with issues of heterogeneity, but there we see strong beta signatures, right? But I didn't show you the spectra from that because we're still working on that.
Okay, thanks a lot.
Okay, so 2 more questions here. Great talk from Ying Jawla. Sorry if this is already in the preprint. Does the combination of electroporation and slow freezing lead to a reduction in the typical DNP enhancement obtained?
It's the combination of electroporation and freezing. Actually, the maximum enhancement that we get, I didn't show you this, but it is in the preprint. The maximum enhancement we get for the electroporated system is a little bit higher than we do for the incubated system. So we usually get around 40 for when we add the amupoba incubation, and we get around 60 for electroporation. But there is definitely, like, it's a slightly longer preparation time, and so there's a little bit more amupole reduction in that time as well. So if we had a more stable amupole, the enhancement could be higher.
Okay, there are 2 other questions here. One's an anonymous one. Says a naive question. Would something like double quantum filtering be helpful to facilitate the labeled background standing out from the natural abundance background?
Yes, of course. Yes.
Mhm.
I mean, it's just a question of sample of experimental efficiency, but yeah, single quantum, double quantum is fantastic, as well as like just, you know, one bond homo and heteronuclear correlations. But yes, of course.
Mhm. Yeah. Gil raised his hand with a question, but it seemed to have disappeared now. Gil, are you there and you want to speak?
Yeah, I just wanted to confirm that Kendra understood my question related to the phosphorus carbon reader measurement. It was meant to actually be a continuation of the question related to the membrane association of the of the synuclein, whether she can actually detect any proximity to the membrane by something like a carbon phosphorus reader or TDOR or
That
any.
would be a delight. I do not have that instrumentation, and I really hope that I know that they were thinking of building that type of probe, a carbon phosphorus probe,
Right.
at the National High Magnetic Field Lab. Yes.
I
Yeah.
think Gert Buntkowski might have one, if I'm not mistaken.
In Germany, in Darmstadt.
In
Okay.
Darmstadt, yes.
Yeah, I would love it. It would be a long experiment, but it would be great.
Well, I mean, I think you can actually retune the existing probes to that configuration.
Carbon, though?
Well, they can be moved around some. I don't know how much, but yeah.
Okay. Yeah, no, that's definitely a possibility. I would love to do it.
I can actually confirm that our 3.2 DNP probe here can do that. I just talked to our technician last week.
Okay, cool.
Yeah,
Well, so
but-
then I'll have to come to France and
Yeah, but it's
Yeah,
broken
you have
now.
two options.
get some croissants.
Okay, Lauren Andreas has a comment. Phoenix NMR has HFXY. So that covers all your bases, right?
Perfect.
All right. Okay, all right. So I think that covers most of our questions. Thanks, everybody, for all the interesting questions and participating in the discussion today. And we would thank again Sudheer for a really interesting talk, telling us how to generate nanosecond pulses at 527 and higher, and Kendra for her insightful discussion of the recherche aspects of synuclein. There are many that we don't really understand yet. It's a very, very complicated system. So Kong, do you have the lineup for 2 weeks from now, for the 15th?
Sure. I mean, 2 weeks from now, we will be hosting Galia Devolocina from UCSD, and then she will be talking about, as what Kendra said, like the way to go is probably to attach the radical to the protein. I think she's going to talk about that, if I'm not mistaken. And then the second speaker will be Klaus Schmidt-Rohr, who will tell us about domain size determination using proton inversion recovery, if I'm not mistaken.
Okay, so we'll have Galia Devolocina and Klaus Schmidt-Rohr on tap for June the 15th. Okay,
Yep.
So thanks again to Kendra and to Sudheer and to all the participants, and we will see you then in 2 weeks' time. Okay, bye.
Bye.
Right.
Bye.